good news, bad news. For those of you who enjoyed it, and I hope most of you enjoyed it, this is bad news. But I hope you will, this is just the beginning, as I mentioned, and you will be, we will be in touch. We would like to hear from you, from especially those of you who are really using those tools and in your research or in your, future work. At this time, today I will there are two topics I'm going to cover. Two components of ProD. One of them is the evolutionary relationships, how we relate sequence relations, correlations to dynamic correlations. And the other is druggability. So I will start with the druggability theory and simulations. Okay. And then continue with analyzing sequence patterns. These are two totally different topics. So it will be between the passage between the two. I may give a break and ask you a few questions. Of course, you are welcome to ask questions anytime. Do you hear me in the back? Is the microphone working? Great. And then after this session, we're going to move to the other one, and Dira will help you apply what you learn to this Druggable component. I didn't give the name, but I like it, Druggable. So it is like a drug graphical user interface, if you wish, but it is mainly for druggability simulations. I think you will like that part because it is, first of all, it's an interesting topic. Secondly, you are using now your MD, everything you've learned from the first part of this course, but now applying it to drug discovery. So we start now. So this is a general overview of the state of postgenomic findings in the area of druggability of target proteins. Overall, we have, after much debate, we converge to this number of about 21,000 genes in the human genome. Now, the thing is that each gene, well, in a very simplistic way, each gene maps to a given protein. We are forgetting the other complicated recombinations, etc. So this means that we have approximately 21,000 target proteins that we may target for curing diseases. Okay? I just started, you didn't miss anything. So this is the druggable genome. We have about 21 genes. The thing is that not all proteins are druggable. That is a new word, perhaps, druggability. But it is exactly reflecting what it means. And experiments, previous work showed that, and these were some data collected from Pfizer originally, because pharma has lots of data, which at this point they shared. Andrew Hopkins was a leading researcher there. So the number of druggable genomes is approximately 3,000. It's a rough estimation. On the other hand, what we know as disease-related genes, we have about 3,000 genes right now also, which have been related to diseases. So there is no point targeting those genes that are not druggable in the first place. It will be a lost cause. And also, we would like to target those that are disease-related. And it turns out that we have actually quite a small set of target proteins. And there is in pharma, some of you who are familiar a little bit, there is this attrition effect, which means when you go to the later phases of discovery, you start eliminating all the initial hits. And this is a really bad thing. So what is recommended now is before you even embark in all those, the pipeline, it's good to first assess whether the target that you are supposedly targeting is druggable or not. So that is one, and here we are going to talk about that. So these were some numbers, actual numbers that have been extrapolated actually. Of course, you know, GPCRs are very important targets, and so are kinases. Now, a simple question. We're going to have fun. This is our target protein. Suppose this is our target protein. Are you familiar with this type of representation, blue, red? Usually blue would be positively charged, red negatively charged, and white would be in between, hydrophobic if you like. So if you look at this structure, this type of surface representation, the question is it druggable or not? Yes? You don't know. Okay. So what are the type of criteria we usually look at? The number one criterion, the most important thing. When you have a drug, the most important number one criterion is shape complementarity. It's all physics, actually, not chemistry so far. So you want the shape of your drug molecule to exactly fit some surface cavity on your target protein, for example. Okay? This is the number one. And, of course, the second thing is chemistry. Physics first, chemistry second. No, nobody gets offended, okay? And my PhD was in physical chemistry, so it's... And my undergraduate was chemical engineering, by the way. I heard somebody, at least, yeah, it's a good starting point. So we have, in this protein, now I'm asking you, do you think it's druggable or not? And if it is, okay, the answer is yes. And where would you think the drug to bind?
Like there are at least 3 potential binding sites. There's large, positive, or negatively
Here?
one. Other side, actually.
Here?
Negatively
Here?
charged. Maybe the binding pocket right there. Looks like a channel.
Yeah, I wish you had a pointer, but...
Yeah.
So here, this one?
Yeah.
Yeah. That's enough. Yes, that is the right answer. Actually, it is the druggable site. You have a very well-defined cavity here, you can see. And that is actually the drug that binds. You see, this is the drug molecule, or in some more familiar representation. And this is exactly where the drug fits. Okay? I remove it, I place it. You see the difference? So that is, we know that this, we can design a drug that will probably fit exactly that cavity. So it's a good thing when the surface presents some, you know, some kinds of nice binding spots. Actually, in kinases have been relatively easy targets because they have those clefts between the different domains. Phosphatases, on the contrary, which are equally important. Kinases will be the phosphorylation. Phosphatases would do the dephosphorylation. Both are needed. But phosphatases have not been good drug targets. And you can easily see, if you look at their shape, they practically present no cavities for something small molecule to bind. It has been a very challenging target. And perhaps it's better to, you know, target other proteins. On the same pathway. By the way, everything is about the function, right? You don't necessarily need to target a certain protein, but anything on the same pathway because you want to interfere with the cellular paths, cellular functions. So these are new concepts that are arising. Polypharmacology, for example, would be using more than one drugs that would be targeting more than one protein on the same target. On the same path, because you want to interfere with the functionality, okay? All right, this is as simple as that. Now you all know this. We look at the drug, and now we know, at the target, we know, oh, it can bind something here. Of course, that's not true. We do lots of expansive simulations. And in this case, actually, this is as you can see, a glycoprotein that is important for intercellular adhesion. And experimentally, it has been observed that a drug binds with nanomolar affinity. Usually, they report these things in terms of the concentration of the drug. Nanomolar is good. It's a strong binding. And when we do calculations, druggability simulations, we even find that, oh, this drug may not even be optimal. You can have something that even binds with higher affinity, lower concentration. Okay? Now, what is the methodology? Good news, the methodology is MD simulations. And we are all now familiar with MD. What's the difference compared to the conventional MD you learned? Well, conventional MD is usually in water, you know. Or perhaps you have the membrane protein embedded in a lipid bilayer. Here, we would like to, we are inspired by experiments, actually. X-ray crystallographers, what they did was to resolve the structure in the presence of small molecules, not necessarily only, you know, and why we try to see where, in the crystal form, where those small molecules will stick on the surface, okay, where they would land. And also by NMR, they did similar experiments trying to understand what are the positions on the surface that are most likely to bind fragments that are drug-like. Okay? So that's what we're doing. So this is the protein. Can you see it? White surface. White on white is a good choice. So this is the protein. And now these are the small ones are the water molecules, which we wanted to de-emphasize. And then here you have a bunch of small molecules. What are they? We have isopropanol, acetamide, isopropylamine, etc. And they have different features. Why did we select this? Well, we were not, by the way, the first to do that. That was another group. But the logic, one logic is that this fragment, isopropanol type of fragment, is the number one most commonly observed functional group in drug-like molecules, existing drugs. So that is, the goal is the following. We're going to do the simulations here, and we're going to see where those fragments, where those small molecules land on the surface. And perhaps there will be some positions that will really like to bind those small molecules. And depending on the type of those fragments, we can even make assessments about which type of functional groups are most likely to bind. Okay? Very simple. So we do those simulations. We see that this region is really very highly populated. And that we start to do some calculations. This is a summary of everything. Now I'm going to go into really much more details and present the methodology in more details. Okay. Also, this is also an example of simulations. So this is cytochrome C. Cytochrome C has a heme group here. And when we do the simulations with those small fragments, which are now shown by those molecules, among those molecules, do you observe one of them that is highly stable? Really, is decisively, it selected a binding pose, one of them. You may have noticed, actually, that there is an Okay, there is actually this imidazole group here. This is an imidazole group, which was one of our selected fragments, which just stuck to the heme group in a vertical, perpendicular position, and stays there. It really likes this position. It won't unbind. It just stays there. And what we are doing is actually we collect a series of snapshots and try to understand what's the probability of occurrence of a given pose, occupancy of a given site. And for sure, that's going to be popping up as a very highly probable binding site for imidazole-like entities. Okay? So these are the type of information that we obtain. Now, let me go into a little bit more details. And the good news is you don't need long simulations for that. Whatever will happen within 10 to 20 nanoseconds. So you start with your system. And of course, we don't know whether this is a binding site or not. Everything is uniformly distributed originally. And then after 10, 20 nanoseconds, we already collect a bunch of snapshots. And we can see that probably this region is more populated. And then we are converting it into some representation. I will explain representation of hotspots, and merging the hotspots to come up with some kind of what is called a pharmacophore model. Do you know what a pharmacophore model is? So if you have a model, you say, okay, if I have something here where there is a negatively charged entity, a positively charged, and the hydrophobic entity is something like that of this shape, then this is likely to bind this region based on the way the small molecules have been accumulating. And this type of so you can construct a three-dimensional model, a positive charge here, a hydrophobic aromatic group here, etc. And then you can screen this model against libraries of small molecules to find whether there are already some drugs that have the same shape or properties. So this type of druggability simulations allow you to build some pharmacophore models. And then you can select, one thing we are doing a lot these days is to screen the FDA-approved drugs. Drug repurposing is like the low-hanging fruit these days. Instead of discovering a new drug, we know that those FDA-approved drugs are not toxic, at least their toxicity is within acceptable limits. If they also serve another purpose, which is called drug repurposing, so it's really good, and we are trying to also understand such cases. Sure.
So the simulation is up to 20 nanoseconds.
Yes.
I understand this is probably very fast to go, but I still assume within 20 nanoseconds, we don't expect to see the conformation change a lot for the protein itself.
That is true. That's a very, very good point. That's a very good point.
So I think what I'm trying to say is, just because thinking about even the binding pocket is pretty, conserved after simulation, then to reach this information, do you think docking can, pretty roughly give us the similar conclusion for that instead of running simulation?
Well, I would think not. You can use autodock also, for example, which is totally unbiased and is. But I think, of course, the docking is a very powerful tool. But we've seen all these simulations, this type of, more elaborate methodologies came up because of the insufficiencies of existing software. So we want to do better than that, and we want our molecule to really, breathe, jiggle, wiggle, whatever you want, in using its own intrinsic dynamics. But the point you made is still a very good one, because within this 20 nanosecond simulation, we're going to have minor conformational changes. Now, if we have a large conformational change that favors the opening of a cavity, this we may not be able to observe. And in this case, a better strategy, and that's what many people are doing, is to have multiple starting points. Again, generate an ensemble of conformers using ProD. And then you can carry out, if you have a well-defined target, you can carry out a series of simulations with different starting conformers. That is possible. Not this one, this one. Okay, here's also a view of what's happening. Normally, when we place a number of these are called solute molecules. You have a solution. In the solution, the solvent in this case is water, and the solute molecules are those fragment-like molecules that we place. Now, if you have those, you start your simulation. The first thing is you heat it up to facilitate the equilibration. You don't want them to cluster anywhere originally, so a more or less uniform distribution. And then this is the annealing, where you have a high temperature, and then you have the equilibration followed by your productive run, what's called the productive run, okay? So originally, everything is uniformly distributed in the medium, in the solution, but then after a while, you obtain clusters on the surface. And you can do those simulations using whichever package you like. Of course, we all like NAMD now. And with NAMD, you can use whichever force field you like, okay? Now, free energy of bind, again, more details a little bit. Now, suppose you are in the equilibration period now, in this red part that I have just shown here. I'm sorry, the productive simulation part. In this productive simulation part, you take multiple snapshots. Okay? You just record the coordinates at multiple. And of course, Druggable is doing all these things for you. But I want you to understand what it is doing. Okay. So you collect snapshots during this simulation, provided that Boltzmann ensemble has been attained. And then the idea is the following. We look at what is called enrichment. What is enrichment of a given molecule at a given location? So a given location is very rich in that molecule, okay? So the idea is to have a metric to assign some kind of binding affinity estimate. We would like to see how much a given type of fragment-like molecule, how much a given location is enriched in this molecule. Should I rephrase it? So we are focusing on, actually we're focusing on, we want to understand what, this is a very, very simple equation. The free energy of binding will be simply proportional to the enrichment. And here, this is the number of, let's suppose we are talking about isopropanols only to simplify. Number of isopropanols observed at a given location on the surface, for example. The density of propanols, if you like, because we divide by the number size. And then we take the total number of isopropanols in the entire simulation. So we know what this would be, the N0 would be what is expected, right? You have a certain number of isopropanol molecules, you have a certain number of grids, or you have your overall surface, which is divided into grids. So this is the expected probability, if you like. But this is, at some points, you have a higher probability. These points are enriched in isopropanol. And this is the enrichment ratio. And if certain positions are more likely to have more than average isopropanol molecules, then these regions have a high affinity for binding isopropanol. And the binding affinity is simply calculated by this simple equation, as simple as that. This is an approximate equation, of course. This is an approximation, but it works well. We don't claim that we are finding the exact binding affinity. Nobody these days can claim that. But we say that we have some estimate, some kind of perhaps optimal, achievable affinity, provided that the system, you have the right binding pose and you have the right small molecule. Okay? So this is actually the most important slide. The whole method is there. So you have a series of snapshots, and each time you count how many times you have a certain location occupied by your fragments, your molecules, normalized with respect to the random or expected value, which is nothing else than the density, original, the input density. Now what happens is the following, again, a little bit more details. The surface of your molecule, actually, you just count, the number of molecules in different grids. What you're doing is to have, let's say in this case, 50,000 grid points. And you are, if at a given position, at a given point, you have a twofold enrichment. So the probability of having an isopropanol there is twice as the overall density. So if you put in this equation, if you put 2 here, ln2 is about 0.69. RT at room temperature is 0.6 kcal per mole. And then you obtain this twofold enrichment gives you a binding affinity of about -4 kcal per mole. Okay? So if you have even higher enrichment, I think this one corresponds to something like 12-fold, 13-fold. You can have an even higher binding affinity, depending on the probabilistic occurrence of this occupancy at this spot. And then you can, you know, can you see it's really very subtle, but we have colors here on these grid points. Some of them are warm, the others are cold. Cold means low affinity, warm means high affinity, okay? So we already mapped the surface and we say, oh, here's a, I'm sorry, here's a high affinity region, here's a, I don't know, low affinity region, whatever, okay? From then on, maybe I'm going too much into details, but it's important to understand at least what's happening. From then on, we start merging things. You know, when you solve a problem, you expand, and then you go back to the solution. This is exactly what we're doing. Now we are going back to have more compact results. So we condense, you know, the hotspots into representative. We condense grid points into representative hotspots, what we call hotspots. So, for example, if there is a very, if there is a warm point here, okay, the lowest delta G value, we consider this as a center, binding center. And then we consider anything that is within 4 angstrom and condense all of them into a unified hotspot like that. Okay? So we now generate a bunch of hotspots. This representation is more, you know, visually or conceptually easier to comprehend, I think. So we have, and you know, the mapping from these original 50,000 grid points to those 191, in this case, hotspots, should obey the same distribution of energy. So it's more or less a reasonable choice in this case. That's what is shown. You don't need to worry about that part. But now what happens is you have this, we identify then, in this region, we have those hotspots, okay? And those hotspots, actually, they are contributed by different, they may be contributed by different types of molecules. And usually, a drug, typical drug-like molecule, there is something called drug-like fragment size. Drug-like molecule will be about 7 hotspots big. So this is the way you obtain, for example, a potential pharmacophore model, okay? One example, so this is what is known from experiments. There is this drug that binds here. And here is what the simulations is showing. I think, so we have this interaction site between MDM2 and P53, which is a well-known binding site for interfering with the interaction between those two proteins. So this one is correctly identified, for example. Or alternatively, what you can do is, again, you have this inhibitor here, and you have your pharmacophore model, if you like, here. And you want to see how well the known experimental inhibitor superimposes on your model, right? And this shows how the hotspot distribution and the known inhibitors agree or don't agree. They don't necessarily always agree, but there are many examples showing us that we're doing well. And we can even, you know, experimentally, this one was 47 nanomolar. By the way, in those type of calculations, if you are at the same order of magnitude, this is an accomplishment, okay? We never try to be within 2, 5, 10%. The same order of magnitude is good. And here they are in the same order of magnitude, but we don't even claim that we have to be. We are more interested in identifying, first of all, whether there's a druggable site, and what would be a probable binding affinity. And then we can elaborate more, you know, with more detailed methods. That's, again, an efficient way. You see how concerned we are about efficiency. This is an efficient way of first screening, if you like, and identifying whether it's worth pursuing it or not. Now, a very important concept. Any questions so far? Because I'm changing topics a little bit. Yeah,
around that.
it has those curvatures, etc. Of course, of course. And we're doing MD simulations after all. You know, you have all the atoms, all the granularity, everything, all the specificities. Now, classical approaches for inhibiting a given enzyme, the classical approach would be to target the catalytic site, right? This is where everything happens, that the enzyme catalyzes a reaction. And if you interfere with the catalytic site, you're going to probably inhibit the chemical reactivity. Fine. But enzymes, most of them, have other functions too. They not only are chemically active, but also they bind other substrates. They are part of a chain of interactions, right? So in addition to those catalytic sites, they have also other spots, other sites that are likely to bind substrates. So it is very important, and these are usually called allosteric molecules. So what happens, there is a region here which binds another molecule, and then you have a response happening here. So there is a allosteric actually comes from the Greek word allo, other, or farther, I don't know. So if you have a binding somewhere here, you have a response at another place. And this has been a feature that has been exploited in drug design, because in some cases the catalytic site may be buried, not easily accessible. But then, I'm sorry, if you target instead the hotspot, which is for substrate binding, that's another way of interfering with the functionality. So these are protein-protein interactions. You now interfere with this protein-protein interaction. This type of drugs are sometimes called blockers, okay, as opposed to inhibitors. So they block the interaction. So the good thing is that the druggability simulations, as you can envision easily, they don't necessarily give us one region which is most likely to bind drugs. We have multiple sites. Some of them are more probable or less probable. But for example, when we apply to this molecule, well, we find this site to be actually very, very druggable. And then there is another one, and then there is another one. But it turns out that this is the allosteric site for this particular molecule. So we can perhaps, it would be a better strategy to interfere with the binding of the substrate. But interestingly, that other site, it also turns out to be the interacting with another molecule, tubulin. So that is also very interesting, because many, many enzymes, they have multiple functional sites. And depending on what functional site you target, you will be interfering with that type of functionality. Okay? I'll give you examples. And this shows the comparison of the hotspots versus what has been experimentally observed, etc. So we have many examples like that. I'm not going to go into the details, but the idea is that the hotspots that are being predicted are essentially agreeing well with what the regions that have been observed to bind the inhibitors. Okay? So that is the major message. And we can identify more than one hotspot. And it is, and I view this as the first step of more extensive, more elaborate analysis, but it's a very good starting point. Oh, these are from some studies that are in progress, actually. I'm not going to go into the details, but it shows an example of where you have, well, experimentally, we know that this region, which is quite buried, actually, between the two dimeric portions of an AMPA receptor, this region binds small molecules, and we can identify in our simulations that it is indeed one of the most probable hotspots. I think I'm going to skip this. But again, the idea is, these are the regions that are being identified. And here, one of them would also agree with experimental observations. So we have lots of this type of analysis. I'll skip these things. So what I have explained so far is, this was yesterday, okay? Flexibility. We talked about the importance of conformational flexibility. Druggability, so we did that. I want to mention a few things about this modularity, promiscuity, which I mentioned a couple of times. So this is what we will do. Now we have the way biomolecules are designed is with the help of modules. They are modular. So it is like if you have a machine, you can plug in a piece here, a piece there, and then overall it functions. And some modular components are actually used in multiple different enzymes. For example, ATPase domain. That is a domain that is being used in many, many different proteins. Or you have SH2 domain, SH3 domain. So that you have different domains, different modular components that are functional and that are being part of different types of proteins. Okay? There are many, many proteins that are ATP regulated, for example. And the thing is the following. If you have a given enzyme and a substrate here, which binds the active site, it is possible that this active site itself is interacting with another site, this type of allosteric effect, remember? So if you target this region, it may have an effect on the catalytic activity. Or you may have multiple modular domains, like shown here, and you may have complexes, like adapter proteins, you know, the way transcription regulation works with the help of cofactors, etc. So you have a whole machinery. And the thing is, I really like this review, which shows the big picture, where you have, in the case of small molecules like that, you have highly integrated catalytic and regulatory functions, very integrated, integration increases. And in this case, as you go to more complex systems, you have separable standardized components. Okay? And these type of systems are more modular. They are also more evolvable. Now, in other words, evolvability, druggability, we are inventing all those words. So they are more evolvable, means they can sequentially, they can vary more. They can adapt to different substrates, different ligands, etc. So it matters which region of your enzyme you are targeting, as I mentioned. So you don't, when you target a protein, actually, you have to worry about the specific site. It's not about just targeting the protein in general, because different regions may have different implications in the cell. Here's a very nice example, actually. You have, in this case, a GPCR. Depending on the it may bind a biased agonist or an agonist. And it would bind at different positions, this one and this one. And then depending on the substrate that it binds, if you go all the way, you have a G protein-dependent pathway that is being activated, or you have an aristin-dependent pathway. These are different functionalities. So your drug may be binding different sites, and they may have different cellular consequences. This is an important concept. This brings us to protein promiscuity. What does it mean? What is it? No selectivity. Okay. It has multiple activities. Let's say, many proteins are involved in multiple pathways. Okay. Depending on the targeted surface region or on the accessible surface, accessible structural change or dynamics. The sentence is not completed. the interactions with different or multiple upstream or downstream partner substrates may be affected. So the protein may have different effects, and which in turn would impact different, may result in different phenotypes. Okay? So the promiscuity is, in a sense, you are trying to, you have many different ways of interpreting, but the protein would be involved simultaneously in multiple functions. You have what is called side effects. Or there is also this concept of drug promiscuity. Your drug, if you look at the existing database of FDA-approved drugs versus targets, we have such databases. For example, Drug Bank maintained by the Canadians. If you think of a matrix where you put here the drugs and the proteins, many drugs would be binding many proteins, and many proteins would be binding many drugs. So this is the promiscuity. You see, there is no one-to-one correspondence. And you can call it in different ways. Each drug has many side effects, you can call, because it's not targeting only one protein. Why? Because they have structural similarities, or they are involved, and yeah, they may be in the same, leading to same phenotype. Sometimes a given drug for a given disease may be effective through different pathways. So that is an important concept. Now I will finish my nice example. Gleevec, some of you are familiar with. When it was first discovered, even Time magazine wrote that that's it, we won the war against cancer. And these are the bullets. Okay? And the drug Gleevec then would be binding, so this is where it binds. It binds tyrosine kinase, BCR-ABL, which is a very important target for curing leukemia. Well, not curing, I would say, but at least dealing with. And this is the way it's coordinated, you know, in more simplistic view. But what happened? What happened then? What happens usually when you have a drug, but after a while the drug is no longer effective? There is this concept of drug resistance. And there may be a resistance to multiple drugs, and it's called multidrug resistance. Okay? So that's what happened, of course. And soon the molecule would mutate a residue, threonine here, into isoleucine, not to allow for the binding of the drug. Okay? Now we have to deal with this problem. We have to redesign the drug to, you know, overcome this blocking effect of this single site point mutation. So this is the well-known resistance that has been observed at. Now, then the same, no, this was Novartis originally. Novartis came up with a slightly modified form. A slightly modified form with the hope that it would be good enough. Of course, it was not. There was the same type of interference, and I'm not going to go into the details. So they used the same scaffold. This is a usual practice, to maintain the scaffold and then change some decorations, if you like. It didn't work. Originally, it was approved. It seemed to work, but it didn't. Again, another drug that fails with the same mutant. To make the long story short, this is one of the most important studies where, now, what was the solution? What would you do? Now you have your target protein, and you are seeing that, you know, your drug, which is binding the catalytic site, is not working. Now, based on my lecture, what would you do? Well, you would try to target another site, right, on the same enzyme. And that's what they did. They targeted an allosteric site, a totally, you know, away position. And here is the image, perhaps. So this is the catalytic site, or the original Gleevec molecule, or the same family of molecules that binds here. But now they have a second drug, which binds this position, okay? And it is the methylation, post-translational modification site, which is important for the functionality of the same enzyme. And now this is like a magic thing. It works. And since I have, the last thing I followed was that it was still going clinical trials. This may have been approved or not, or failed again, but at least that was a good strategy. Okay. And I wanted to finish with an example from our work and emphasize the importance of pathways. So I don't know how much you are familiar with this type of pathways, but this is a fibroblast growth factor, which would be triggering a cell growth pathway. Okay? Now, and there are kinases involved, etc. So it goes all the way to the DNA. Now, one of the components here is a DOSP6. This stands for dual specificity phosphatase 6, a bad target on which we spent some time. And then for this, what the phosphatase does is to actually inhibit that pathway. This is the symbol for inhibition. Inhibition means it won't allow the cell growth, okay? So if we have a drug that won't allow, that inhibits that phosphatase, what is going to do? What is it going to do? It will accelerate the growth, right? And so that's what actually we did using a zebrafish embryo. And well, this was again the work of Ahmet, by the way, who was one of the first authors. This is always like that. If you're doing computations and you collaborate with experimentalists, you may be the first author like that. One of the first authors and your name. But we're fine with it, even being recognized that our contributions is equally important, at least. Anyway, we have, he did these simulations, which I thought it was working. Oh, yeah. And he was able to show where this small molecule will bind, and then it will interfere not with the active site, but with the allosteric site. So that was one important finding. And this was a big project. That is what happens. One of them is doing the experiments with animals, the other one the cell-based, and Ahmet was doing the in silico, in vivo, in vitro, we call it, all three together. And then it was, we were able to show that the cardiac lineage underwent an increase in size. So the growth was, if you want to grow things, that might be a good strategy. In general, you want to shrink tumors, for example. That would be really bad in this case. So anyway, I want to finish with a nice, let me go backwards. I think, Tim, you prepared this slide. Is that true? This is, I have to acknowledge Tim for this beautiful slide. And this is our conclusion of this part of the lecture. So in principle, I'm sure Tim can explain better than me, but this is my version. We have, you know how these days the imaging technology is becoming very, very important. People are extracting all information from images, from high throughput or high content screens. High content screens would not be high throughput, but providing more information at each well. So this type of data are being analyzed to infer some statistical information. And then to make inferences about pathways. So what are, for example, the pairs of proteins that are co-evolving? Then they ought to be connected somehow. Correlation, we have learned yesterday, we have this connectivity map, remember? The inverse of it was the covariance. So if two things covary, then they also are connected. So, and the connectivity map, you can invert it to find the covariance. The covariance, you can invert it to find the connectivity map. So these data give information on covariance, and then you can go into paths. And then you can identify some targets, elaborate on those targets. And of course, you can perform simulations at the cellular level even, not only the target, the molecular level. So this looks like the surface of a molecule, right? It's not actually. It is a cell, a snapshot from a cell surface. And these points here are individual molecules. And then you compare with experiments, and this is a cycle until, and you, of course, I mentioned drug bank already. We learn a lot from existing databases of structures. We use this information until we are satisfied to move to clinical trials. This completes that section. Now we have time for questions. And then I will completely switch gears. I need a question from the back. Then you will be next. Someone from, I'm sure you have questions in the back. Yes.
So you mentioned shape complementarity being important.
Number one, yeah.
So where would you rank electrostatic complementarity in your experience?
It would be next, to my view. Yeah, you need to fit the shape first, because it's like, hardcore interaction. The shape is very important. But now the shape fits well, but here you have a hydrophobic surface versus a charged polar surface. This is not good. Then you need to see whether the complementary, electrostatic is extremely important. But first thing is shape complementarity. Oh, many questions from the back now, but she's next.
Thank you. So at the beginning of identify the hotspot or define the druggability of a protein, you choose several small fragments.
That's correct.
Probe molecule.
These are probes. Very good. I didn't use the word. This is the right word. These are the probe molecules. Probe molecules.
I understand people try to use even smaller, like water or methanol or something, but you use this different things, and the reason for that I'm trying to understand is because it has been commonly used in the drug formulation.
Exactly. We have the statistical data. We analyze all the existing drugs and try to identify what are the most frequently occurring functional groups. So this isopropanol group is the number one, most common. Yeah. And then, we go down the list. In some cases, now, right now, we are, Indira will perhaps talk more on that, but we are increasingly selecting a broader group of drug-like fragments. Of course, this is one of the areas that still needs some improvement. But originally, the first study in this field was done with those 4 molecules that I've shown. In the next study, we used a larger data set. And we don't want to do a very, very large one because that would be competition after all. Yeah.
So I guess my question really is, you use this probe to pointing the key interactions, right? And this isopropanol, what do you think? What kind of information are you trying to get? Hydrogen bond donor receptor, or just the size?
In this case, it is mostly hydrophobic, actually. Most of the isopropanol molecule would be hydrophobic, and then there's this OH, polar, slightly polar group. Yeah. Yes.
Wouldn't it be possible to get an estimate of the error of your approximate delta G based on the finite size of the system?
This is an excellent question.
I mean, certainly you could run multiple trajectories.
Yeah, that is true.
Error bars, but
yeah. Yeah, that probably. Yeah, we didn't do that, but that might be, maybe carrying out independent runs and then seeing what is the variation in the delta G. Yeah, that is possible. We haven't done that. And we have been avoiding making big statements about the numbers, and we have presented our results as the upper limit, the strongest binding affinity, because we are, in a sense, optimizing the conditions, letting them bind. So we refrained from, elaborating much. This is the average value. This is the error bar. We don't want that. We're very happy right now with the more qualitative results, whether a given site is druggable or not, whether it's a high affinity or low affinity binding spot. But yeah, it's always possible to be more precise. But
as a follow-up, as opposed to the simulated annealing that you're currently doing, have you done this with, say, replica exchange? Where you can run 20 simulations at different temperature and swap them across boards?
yeah, that's possible. That's
And
possible.
look at the equilibrium.
That might be in the next extension. We can work together on doing that. It's always possible. Yeah, or there are other methods to to have better sampling in MD simulations. You can have accelerated MD. You can analyze the trajectories using different methods, weighted ensemble, etc. But yeah, everything is possible. You can. This is like the basic framework that we have, and you can always improve it. That was one question over there, but I think it.
I have an obvious question, so there's probably going to seem silly to the group, but
There is no silly question.
so to model the drug binding pockets, do you have to have the type of information we found yesterday in terms of like the flexibility?
Yeah.
So you have to have, so for those you have to start with things that have been solved with an NMR.
Yeah, that is also something we discussed in the very beginning. Now we're taking the X-ray structure in those simulations, but and these are very short simulations. So, and these cases that we studied were, perhaps easy cases, which were able to bind a hotspot, a drug, without a significant major change in their conformation. But if there are these large-scale movements, a better strategy, and many people are doing that, is to start with multiple rounds with a nice distribution of starting conformers. Okay. There were a few more hands here, which disappeared. This was you, okay.
I was curious about the MD simulations. Are you considering full atomic or
Full atomic, yeah.
coarse-grained?
This is full. This is conventional MD, what Klaus described, yeah. All right, no more questions. Now we are moving to another field. How many of you have some bioinformatics background or interest? Interest, yes. Okay. All right, so for those of you who have this bioinformatic background, it might be trivial, but still. This is my lecture 4. Where is the lecture 2? You will ask. Lecture 2 was Tim's. All right. I showed you this slide. Some of you will remember it. And I'm showing it again. Not because I'm really very impressed by the work of Don Tevrik at Weizmann Institute, but also it shows an important concept, that of substrate selectivity. Yesterday I mentioned how the given protein may change shape depending on the different bound ligand. But it may also, this type of conformational flexibility is also accompanied by some sequence variability, okay? To facilitate, to optimize the binding, and to select actually a given substrate, you need also some kind of sequence variability or some, sequence coevolution in many cases. So that sentence, I think this is an important sentence. Conformational flexibility and sequence variability mediate substrate selectivity. So when we do our analysis, we have to take account of this type of both components. And now we are proceeding to what is sequence variability. Okay? So one of you who was who said he knows bioinformatics left. Maybe I discouraged him. Maybe he doesn't need to. That's fine. Now here we have a, suppose you have a given Gantt query sequence, and now you are looking at an ensemble of sequences that share many common amino acids. So these may be either orthologs. So this could be orthologs, means this is the same enzyme but belonging to a different species, human versus mouse versus whatever, bacteria. Or these may be paralogs in the same species but similar functionality, things that have been changing throughout evolution. Usually we do the analysis with orthologs, different species, but you can have either. Or you can have the same species, same enzyme, same protein, but mutants, okay? You can, you align the sequences. So this is your first sequence, the second sequence, third sequence. This is called a multiple sequence alignment, right? And in the multiple sequence alignment, the whole point is to look at columns. First of all, the correct alignment of sequences from different species, from orthologs, whatever, is an art in itself. But suppose you align them, and in some cases you don't need to do, because some databases, like PFAM, which Prodi is using, which Evol is using, already provide you with some optimal alignments, given your, the degree of, depending the family relatedness you want to include. You can have bigger multiple sequence alignments or smaller. You can have fewer sequences or more sequences. Now, suppose now you have your alignment. The next thing we do is to look at columns. What has been aligned? See what happens here. We have all the way only valence. So this position, these are different positions, I plus 1. This position is fully conserved. And then we have here, let's focus on I and I plus 5. Apparently, this is an interaction. Whenever R changes to E, the E changes to K. What does it mean? You need a basic information. This is positively charged. This is negatively charged, right? Then you have a negatively charged. It mutated in this particular sequence to E, and maybe not mutation. It's another species. And then here it became K. So this tells us that, and the trend continues, positively charged versus negatively charged. Each time there is a change in one of them, the other one also undergoes a compensating change, actually. It's called compensating mutations, correlated mutations. And these are co-evolving pairs of residues. They evolve together, okay? Why do they co-evolve? What do you think? Why would they co-evolve? So this was a positively charged negative. Now this became negative. This became positive. Why?
Maintain charge distribution in the protein.
Charge distribution would be a very, how to say, global property. It's more local than that usually.
So they'd be interacting, so then you retain the interaction by the split.
Yes, indeed. Indeed, that is the desirable reason, let's say. So what we may infer is that, when this becomes negative and this becomes positive, perhaps these two were interacting, a positive versus negative charge. And when by, those mutations, they happen all the time. But this won't be sustainable unless its interacting partner becomes now positively charged, okay? So these are compensating mutations. So ideally, if we have those co-evolving pairs, and then if we have the structural data and we see that, oh, these are really making contacts, now we are really happy. We understand why. In many cases, we don't. In many cases, we don't. And people have been interpreting the coevolutionary data in the context of tertiary contacts all the time. So if two amino acids co-evolve, they are likely to make a contact in the structure. That's what they think. But nowadays, we are starting to think a little bit more broad, flexibly, let's say. We are thinking that not only interacting residue pairs that make contacts may co-evolve, but there may be some more allosteric effects. So something happening here may have a consequence elsewhere. So we are trying to analyze because many of those signals that we detect are not necessarily contact-making signals. By the way, did you need a break to go out? You did. You want to take a break? Okay, let's do 5 minutes break, sorry. 5 minutes. And we are coming to the most exciting place. Please don't disappear. I'm sorry, I didn't think that you might need a break.